Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
I would like to introduce to you Anya Brickman-Reardon, who is the current president of AMITA, and she will be sharing a short welcome and will be saying a few words about Ellen Swallow-Richards and her work.
Hi, everyone. Thank you, and sorry for being a few minutes late. I am juggling childcare today, so I will also probably disappear after a little bit. Apologies in advance to our wonderful speaker. But I did want to welcome everyone. This is an important, not only is it one of our regular annual meetings that we at AMITA hold, but we want to honor Ellen Swallow-Richards on her birthday. And I'd also like to honor past presidents of AMITA. So if you're in the room, please stand up. If you're online, please raise your hand, either virtually or in real life. Just want to take a moment and thank everyone for their service to AMITA and in keeping one of Ellen's legacies alive and going forward in supporting women, both students and alumna from MIT. And then just a little bit about, we try when we celebrate her birthday, in addition to the cake that I hope everyone enjoyed in person there, to carry forward her legacy through ideas that have evolved and developed based off of, in some way, what she worked on or related to the things that she's worked on. So in the past, we've talked about some of her work around nutrition and the ways that's carried forward. And this year we wanted to focus on sanitation engineering and where that has evolved. I am an MCP, a Course 11 graduate alumna. And so a little bit close to my heart here, it turns out that Course 11 was originally sanitary engineering at MIT. And that was inspired and developed by Ellen and her work related to sanitation and engineering and how to make our cities and our communities healthier places. Certainly, this is something that's all been on our minds for the past several years. If we didn't think about it before, we're certainly thinking about it now. Many people thought about it before, but now it's personal. And so I am delighted to carry that conversation forward with the latest technology and innovations in understanding community health through our waste systems. A good topic for after enjoying ice cream and cake. It's why we did ice cream and cake first rather than after today's discussion. But just wanted to welcome everyone, and I hope you have a great time meeting each other, networking, and learning about Biobot.
Thank you, Anya, for that introduction. So a couple of housekeeping things before we get started. I want to let everyone know that this is being recorded. So just you'll have to, if you're online, you'll have to give permission for that. And if you're in the room, you should know that that's happening. And second, so I'm going to do the introduction and Mariana will speak. Then we will have a moderated Q&A. But after the Q&A, we're also going to have a few announcements from Colleen Smith. So stick around. And those of us who are here in person, I am actually here in person. I'm just in a different room. I'll see you guys after the online events. If you wish, you will be able to get a tour of the Cheney room. And Colleen will tell you more about that, but I'm just letting you know so that if you're here, you can think about sticking around till the end. All right. Now to get into the meat of things, it is my great pleasure to introduce to you Mariana Matus, who is the CEO and co-founder of Biobot Analytics. She is originally from Mexico City, and she received her PhD from the Department of Biological Engineering at MIT and in computational biology, where she specializes in the emerging field of wastewater epidemiology. So in addition to her doctoral studies, she co-founded the MIT Underworld Smart Sewers Project, leading the vision and funding for a $4 million interdisciplinary research project with collaborators from MIT's Department of Urban Studies and Planning. And this work led to the creation of Biobot Analytics and set the scientific foundation for Biobot's wastewater platform, which is now serving the CDC and communities across all 50 states. She also plans to expand Biobot's venture-backed platform, which has secured $30 million in funding into countries all over the globe. She has been recognized for her leadership and entrepreneurial excellence through several awards. In 2020, she was named to the CNEN Trailblazing Women in Chemistry. She made Newsweek's list of America's 50 Greatest Disruptors in 2021 and their list of America's Enterprising Idealists in 2022. Boston Globe named Dr. Matus number 8 on their inaugural 50 Tech Power Players list for 2022. And most recently, she was honored to be featured in the 2022 Time 100 Next list as an industry innovator. So, wow, that's a lot of accomplishments. So I will turn the stage over to you, Mariana. Thank you for being here.
Thank you, Julie, and thank you everybody for today. Similar to what was said in the beginning, I have my daughter with me here right now, a 2-year-old daughter. So in advance, my apologies if at some point my talk gets a little bit disrupted by her. I hope that she continues to be busy and entertained by the TV a little longer while I'm on this talk. But yeah, I prepared some slides to share more about my journey from a PhD student into Biobot, now a co-founder and CEO of Biobot, to share about some of that research that kind of fueled the founding of the company. And I have a lot of putting together this deck and kind of realizing just how much has happened already in the past few years. So I'm going to start sharing my screen, and then we'll have time at the end to do a Q&A. So I think I'll just kind of aim to go through the slides for now. So maybe before diving into Biobot and kind of that founding story, I want to just take a few minutes to talk about where I was before MIT. I came here to do my PhD, and I'm originally from Mexico City. I did college at the Universidad Nacional Autónoma de Mexico, UNAM, which is the kind of the top public university in the country. And then from there, I was supported via a fellowship from the MIT, from the Mexican government to go to the Netherlands. I went to Wageningen University to do a master's in environmental biotechnology, because even back then, I was already just the wastewater industry. Come here. This happened earlier than expected, but my daughter's here. So I went to the Netherlands to study wastewater and to understand kind of the state of the field and how can I apply my expertise, which even since college, I was always excited about bioinformatics, computational biology. So like that interdisciplinary collaboration between biologists and data scientists in order to better understand life. I also got to spend a few months in the University of Cambridge as an intern, and then finally made my way to this Cambridge in order to study at MIT. And I joined the lab of Professor Eric Ohm in the biological engineering department as part of the computational biology program. It was there that I was very lucky to have an advisor that was very encouraging of my ambition to go into wastewater, even though he didn't have at the time any sort of like research into wastewater. But one day he just came back to me. He had just met Professor Carlo Ratti at the Department of Urban Studies and Planning. And he came back to the lab to pose a very crazy idea. He said, why don't we look for flu in the sewers? now that we think about it, we are a lab that studies the gut microbiome. We're used to thinking about poop as data. What happens if we look at our collective microbiome and, we start to look for some public health indicators? Can that work? I was a first-year PhD student. This was 2013, so almost 10 years ago. And for me, it was like light bulb moment. I was like, oh my God, I need to do that. Like, that sounds amazing. And we should be doing flu and maybe many other things. Just imagine the possibilities, because we are shedding bacteria, viruses, metabolites via our waste. So I became very enamored of the day one, but there weren't many takers. Actually, most people in my lab thought it was a cool idea, maybe a funny idea, but not really something where they wanted to dedicate, their PhD or their postdoc work to it. It seems a little bit like too pie in the sky type of project to be taken seriously. But I had a fellow PhD student, first-year PhD student, that actually also started to work with me on this, Chris Bandoro from the Ron Sadler lab. And we just got, in collaboration with Sensible, with the Ron Sadler lab, we began this project looking at wastewater from the Back Bay. You can see here actually the location of the manhole that the city was kind to open for us. And we didn't really ask any questions. We were just like, okay, the city offered this manhole, let's go and work there. And then we started to collect samples every Wednesday, through basically every Wednesday through the entire winter season that year. Again, at the question, our objective was, can we track flu cases via the wastewater? So we were imagining this plot in which we could track the amount of flu in the sewers, and we could also track the number of flu cases over time, and that we would see a very clear correlation between the two of them. The reality was not like that at all. We failed miserably. This was that flu season here in Boston. So at the bottom, you can see the flu-like illness that the city monitors and gathers this data kind of on their own. This is the flu view from the CDC. You can see here that winter flu spiked kind of in early January. And then you can even see the colors indicate the different strains of the flu. I cannot read it here, but if it's flu A or B or H1N1. But anyways, and then the above plot, that's my data, where every line is like a sampling event. And then we were only able to detect flu in one sample in October and one sample in early March. So it wasn't even like, oh, well, actually in January, we couldn't even sample because we had so much snow that year that the city didn't want to go and open the manhole for us. They said, we're too busy doing other things, cleaning snow. So we couldn't sample in any of the peak days. And then the detections that we saw didn't seem to make sense either. They really seemed like random. At that point, we had so many questions. Okay, clearly we were able to detect flu, but it was nothing like what we expected. What affects sensitivity of detection in wastewater? How do we select the sites to collect wastewater from? The city volunteered that manhole, but how many people were represented in that particular manhole and those wastewater samples? What is the best time of the day to collect a wastewater sample? We were there every Wednesday at 9 a.m. and we were like, oh, well, actually, maybe that doesn't make sense because maybe most people are gone by then to school, to work. And what volume of wastewater should we be collecting and testing? And is the flu the best use case of wastewater as a public health platform? What has it been used before? What types of human health biomarkers can we identify besides the flu? And what are the most relevant laboratory and data analysis techniques? So when I like something, I don't usually give up easily. So I could have really dropped the project and just moved on to something else, but I decided to actually use that failed pilot to write a grant. and to design a system. If you know, my PhD was in computational and systems biology, so I was used to thinking about more like, okay, this is really like a systems approach is needed, from where do we collect the sample, how do we test it in the lab, how do we test it post lab, you know, kind of to begin to really think about all of the pieces. And we designed the Underworld project, a project where we were going to be exploring progress on mapping the sewage network to understand flow and understand the pipe connections and where we should be sampling to begin with. How to collect the samples better. We actually set up a plan to develop new hardware to collect wastewater samples in a more scalable way. Again, explore different types of biological and chemical assays to look at the viral, bacterial, and chemical fingerprints of wastewater. Understand what other data we need. Like, we probably should be looking at census data to at least understand how many people are represented in the sample, but also ideally what type of population is there. And finally, how do we visualize that? How do we present it so it's helpful for stakeholders other than scientists? So that project got funded for $4 million via the Kuwait government. And it was a big collaboration between 6 labs at MIT, led particularly by Professor Eric and Professor Kalorati, but also with participation from other labs, as well as researchers from Kuwait. It was a big project. It became the topic of my PhD dissertation to work on it more full-time. And now I will tell you about one project that we did under this grant. There were many other projects, but this one in particular I like to talk about because it really influenced me and set the stage for why I decided to found Biobot. One second. I'm gonna call my husband to comfort Lily. Okay, hopefully my husband heard me with that. Okay, come here for a second, baby. Mommy. Of all of the labs that were involved, I would say my lab was one where there was the most question marks and skepticism about the potential success of this project, because in plain words, other scientists in my lab kept asking me, well, I can imagine that there's biomarkers present in the wastewater that are coming from pee and poo. I can imagine that there are, bacteria and viruses that maybe live in the sewers because that's sort of like their home and that's their environment. But what I cannot imagine is that you can actually get accurate data that reflects what's coming from people, what's happening in people. And very frankly, like, is it all just like noise? Is it just noise? Similar to that first flu study where clearly the 2 samples that were positive sort of like didn't make any sense with like what was happening with the population. Is it just noise, or is there, and a way to ask this question was, okay, can we try to understand what's the % of information in wastewater that's coming from people versus like that is present in the wastewater environment, like that is part of like the sewer ecosystem. So in order to answer that question, that's kind of the purpose of this project. And again, then, okay, how do we advance that question? Then we decided, okay, to ask what % of biomarkers present in the wastewater correlate with the diurnal cycle of water. It's very well known that how we use water at home. Basically, this is data that is very reproducible, where if you plot time, like here, time on the x-axis versus water use on the y-axis, basically there's very little water usage at night because we're sleeping. started, here's midnights, and then this is 11 p.m. So if we look at the nighttime, there is very little water usage because we're sleeping. And then water usage spikes in the morning, in this case, between 7 and 8 a.m. during the weekdays, and then a little later during the weekends. And then it stays quite stable throughout the day with a little bit of a spike before bedtime. So we set to then collect wastewater through a diurnal cycle, like every hour through 24 consecutive hours, and to collect untargeted bacterial and chemical data to try to understand this question. This time we were very careful with our experimental design. We collected the sewer network data and associated flow from the city of Cambridge. Cambridge is one of the cities, by the way, that makes this data very publicly available, fortunately. In many other cities, this type of maps are highly restricted, but Cambridge, fortunately, is very good with open data. So we were able to get this data, get all of the manhole locations, get land use data, get demographic data, and then using ArcGIS, we were able to basically understand exactly which manhole to open to capture a highly residential area in Cambridge near Central Square. As you can see here, that catchment has over 80% residential land use. And then we also knew that the travel time of wastewater up to our sampling location was very short. On average was less than 10, 12 minutes, as you can see here. So we also knew that the sample was very fresh. And that could be good because to kind of remove any potential breakdown of molecules or any sort of like noise from that. So we were very careful. This time it wasn't just like, oh, the city opened a manhole for us and we knew nothing about it. We knew exactly which manhole. It also had the advantage that it was located across the street from my lab, which was good because we needed to be transporting all of that wastewater back to the lab to be processed pretty much in real time. Then, as I mentioned, we collected a sample every hour, a grab sample every hour over 24 hours, which meant I couldn't do that on my own. I had to train a group of 30 researchers, all of them volunteers, to come and help me out. I had to train them on not just the protocols I had a team in the manhole, and then I had a team in the lab, and they had to be trained to do either collection or processing. And I had to train them also on all of the biosafety, because wastewater can be dangerous for people if you don't know what you're doing with it. And then on those 24 discrete samples, we basically produced untargeted mass spectrometry data, as well as 16S DNA sequencing data, in order to identify a list of metabolites and a list of bacterial units in order to do our untargeted analysis. That was also really cool, by the way, because all of my experience up to this moment had been with more like microbiology data, like sequencing data, but I got an opportunity to work with mass spectrometry data, and that was just like very cool. And it's something that we still carry to this day at Biobot, is the ability to do not just kind of molecular biology, but also chemistry work. And so once we had all of this data, the first thing that we did was to look at do a correlation analysis of the wastewater microbiome and metabolome. We measured over almost 4,000 different metabolites in the wastewater, and we measured over 250 bacterial OTUs in the wastewater. And then we ran a correlation analysis, and basically what we found were four interesting clusters, kind of what we called an O1 cluster and an O2 cluster for bacteria, and then an M1 cluster and M2 cluster for metabolites. And I will tell you more about those clusters just in the next few slides. When we look at the metabolite content in wastewater, we found that the vast majority of metabolites that we measure, almost 80% of them, correlated positively with the wastewater diurnal cycle. So I'm showing here on the x-axis is time, again, starting at midnight and ending at 11 p.m. And then the first panel shows the flow rate that we measured at the manhole where we collected our samples. I collected data from that manhole over about like 30 different days. So here you can see kind of the average and the standard deviation from that. And just as expected, it followed that known kind of diurnal pattern of going down at night and then spike in the morning, very stable, and a little bit spike at night. The second panel shows you kind of, this is a total amount of metabolites measured at the different time points. So obviously just very high level, first of all, obviously we see that at night, there's much less metabolite content in the wastewater than during the day. And that makes sense, and that's great. But also secondly, if we color the metabolites that are part of the M1 cluster versus M2, very clearly we can distinguish how the M1 cluster is capturing the metabolites that have that diurnal cycle that basically decrease at night and then come back in the morning, versus the M2 cluster are metabolites that are stable throughout the entire day, as you can see here. And more likely they are coming actually from just the sewer environment that doesn't change. We were also able to identify specific urinary metabolites, like you can see here 2 examples, APG and urobilinogen. And they both also follow kind of that trend of decreasing at night and then being present during the day. And we were able to capture fecal metabolites, including some bile acids. And in their case, their dynamic was even more extreme, with basically almost 0 at night. And then during the day, instead of having like a constant amount, we saw this big spike at 8 in the morning, another spike at 3 p.m. So, you know, this was obviously, we only observed one day. So we cannot for sure answer this question, but this seems to indicate that there's a very strong peak of pooping at 8 in the morning, which was, you know, it was just so cool, and that we were almost wanting to go back and do this experiment a few more times to double check that, but it was a lot of work just to double check that particular hypothesis. So we kind of left it there for now as a hypothesis. If we look at the bacteria, we found that 40 to 80% of the sewer microbiome correlates with the diurnal cycle, again, depending on the time of the day. And so you can see with the bacteria and the sequencing data, it's kind of harder to do total quantification. So we can only really do relative quantification, which is why these bars, kind of what's interesting to see here is that the gray cluster, kind of this dark gray, are the bacterial communities that are sort of like probably like part of the sewer environment and ecosystem. And then the orange cluster are the bacteria that are coming from people. And in this case, it looks as if the environmental component goes up at night, but really probably it's an indication of the human component going down at night. Now, if we look at the composition of the bacterial clusters, the one that correlates with the diurnal cycle contains Firmicutes, Bacteroidetes, which are bacteria that are classically dominant in human poop. And then if we look at the sort of like cluster O2, which is the one that seems to be the environmental component, that one is dominated by proteobacteria, which kind of also makes sense, that pink color. And yeah, this is another view of that same data, but if we look at poo, kind of this bar here indicates what the top bacterial families look like in human poop. This is the average of hundreds of people that have been sequenced, well, that their poop has been sequenced as part of the Human Microbiome Project. And then this is how the O1 component of wastewater, the kind of that human contribution component of wastewater looks like. And then that is how the environmental component looks like. So overall, like a pretty good way actually to assign source to the bacteria. So coming out of this project, I was absolutely fascinated and convinced that the vast majority of metabolic and bacterial biomarkers in wastewater are coming from people, and that there are trends that make sense, that this wasn't just noise, that this was actually a trove, a treasure, a trove of information waiting to be understood. And that to me was what gave me the conviction that I wanted to create an organization that was full-time dedicated to understanding wastewater as a source of public health data. I was on the trenches there for many years as a PhD student, so I knew that it would be hard to achieve scale and the potential of the platform from within academia. There was just a very big amount of operational logistics in order to get the wastewater and get, kind of the buy-in from the cities, all of that stuff. So I became convinced that it needed to be an institution of its own. And that's what kind of propelled the founding of Biobot, where we said at the high level, what we want is to collect wastewater intelligence to deliver untapped insights on human health and human behavior. If you want to read a bit more about that founding moment and kind of the decision-making process there, HBS wrote a case study on that moment about Biobot that is publicly available. And the TLDR is when my co-founder Nusha Gailey, who was part of the Underworlds team, one of my research collaborators, and I, when we both decided to form the company, both our professors at MIT, Eric and Carlo Ratti, were supportive, but at the same time, they were right in saying, you don't really have a specific product idea here, and I don't understand how this can be a company, right? Like, isn't this just more research that should be happening in academia? Why don't you stay here? We get more grants. They were obviously very good at giving us ownership of the project and, really being kind of championing us to do more of this work. And even with that very strong feedback, both Nusha and I decided, like, no, we want to create a company. This should be a company because we need to attract the top talent. There's again a lot of operations that don't quite fit the model of academia. We need buy-in from governments. All of that mean it needs to be a thing of its own. So we went for it, even if we really didn't have a specific product yet to go on and get customers or investment. Before going more into Biobot, I want to just quickly mention that in 2017, I was awarded one of the, I was part of the Graduate Women of Excellence Award. And this is a picture of that time with my co-founder, as well as Professor Liz Kujawinski, who works at WHOI, the Woods Hole Oceanographic Institute, and who was our collaborator and mentor for all of the chemistry work. And of course, at some point, I successfully defended my PhD. This was in mid-2018, and I just had to share a picture of the cake because, I don't know, when I found it, I was like, oh my God, I need to kind of bring it back because that was, I was very much, I remember I was so tired around then because I had been trying to balance the early steps of the company with writing my thesis and kind of preparing for my defense. And I was like very, very tired. I think it was one of the times that I've been the most absolutely exhausted in my entire life. But, again, with the support of everybody, it worked out. And my husband is the one that designed this cake, which was just really, really nice. But yeah, now to kind of switch gears to talk about Biobot. So Nusha and she became my collaborator in 2014. So, she's been on this also for now 8 years or so. I am a computational biologist. She is an architect. I am Mexican. She is Canadian-Iranian. And we met both at MIT. She was in the Sensible City Lab in DASP under Carlo Ratti, and I was with Eric. And I think that we really melded very well, despite having such a different background, because we both were curious people that like to work at the intersection of disciplines, right? Like we were not just like, I don't know, just a biologist in the lab or just an architect in the designing a building. It was more like she was interested in smart cities and science and technology, and I was already working at the intersection of data science and biology and chemistry and virology. And I think like we both really enjoy that kind of intellectual challenge of working at the intersection of disciplines. And also we both had a very global mindset. We both had spent time in Europe. We both had moved to the US. So we were very comfortable thinking about like this global platform. And she was also part of the Time 100 Next award list. And, you know, we've really grown to become global thought leaders for wastewater intelligence. We're evangelizing this new space. That's very important because this space was very niche. I remember when I was doing my PhD, people thought it was a great novelty, but it was not very mainstream, even within academia, let alone kind of it being a mainstream market. And we've been very successful at kind of fostering that and championing that, this into growing to be more mainstream. As mentioned in the intro, we have raised almost $30 million of venture capital to date. Right now, we're raising a Series B. You can see here some of our investors. We are also a Y Combinator company, which is a very selective and exclusive accelerator based in the Bay Area. We were there in 2018, and it was together with all of the entrepreneurship programs that MIT that we were part at MIT, like the MIT Delta V, Ideas Global Challenge, Sandbox, DesignX. It was those programs and YC that really allowed us to begin to think as like business people and entrepreneurs, even if we both were like basically very technical in our formation. And now like it's kind of crazy to see how far we've gotten from like that one site and manhole that I was talking about in Cambridge. Now we are working in over 600 sites and still actively growing. We estimate there's around 40,000 sites that make sense to kind of be testing as sentinel sites here in the US, and we are, you know, in 600. So we still have like ways to go, but it's super exciting. And to see like that geographic spread and representation is just really, really exciting. You can see here that coverage. And then we've finally been able to make the type of plot that I imagined 10 years ago, right? It isn't for flu, it was for COVID. But we finally can make a plot. In this case, this is actually a nationwide plot that we calculate based on all of the sites that we cover, where we are showing the level of SARS-CoV-2 virus in the wastewater in blue, and then the reported COVID-19 cases in green. And we can see that there's times of the pandemic where both the wastewater and the clinical data correlate very well, like here. And then there's been times in the pandemic where they kind of don't match, like early on in March of 2020, or lately, they don't quite match. And that's because the wastewater data has very good representation and coverage from people because we are all using the toilet. But clinical data, mass clinical data was something that we did all in some part of the pandemic, but we're not really going to get PCR tested anymore. And then in the beginning of the pandemic, those services didn't exist either. So you can see here, that's obviously one of the huge advantage of why government wants to continue doing wastewater epidemiology, is because that data, they can continue to understand the true level of disease activity, regardless of the availability of clinical testing and regardless of people needing to go on take time out of their days or even pay for getting a PCR test. I should add that this level of coverage is possible thanks to this partnership that we have with the CDC News team. Thanks to them, they are one of our biggest partnerships, and the fact that they are supporting this work to happen nationwide is just huge. And it's very exciting to see that the CDC was able to move fast enough to adopt this new type of data in only in less than 2 years, basically, because 2 years ago in 2020, there was nobody saying in government, I need wastewater data, right? That was not a thing. But now they see the value and they want to continue doing it. And not just for COVID, but for other things. So we're really hopeful that, especially having a partner like Biobot, that they can make that a reality, that they can keep receiving this data going forward, and again, for a variety of different public health data. I should add that we are also sequencing the wastewater to be able to look at the variants that are circulating in our community. So you can see here an example of the different variants that we are looking for and kind of how they distribute regionally. And just this upcoming week, we're gonna launch a time series view where anybody can navigate how the variants have changed throughout the pandemic and how the dominant variants have changed throughout the pandemic. So far, we've only usually shown the last data point. And our vision, again, is more generally now the big transition that we're going as a company is to begin to serve to our customers wastewater intelligence as a more broad data service. So not just COVID and variants, but now we're also doing analysis for monkeypox. We are doing analysis for high-risk substances, including methamphetamines, cocaine, opioids. And we are going to be launching a bunch of new other applications in 2023, including flu and RSV, Hep C, and others. So I should add that for flu has been actually a very active area of research for us at the company. And I should add that, to be kind to my younger self, I should add that flu is the hardest application that we've tried to build on the platform. The virus seems to be very unstable outside the human body, and you need way more sensitivity in order to get it. But we're going to be launching it. It's possible, so we're going to be launching it in the spring, finally available to our customers, not just through pilots. And the other ones, actually were much easier than one would have expected, again, with the origin of the flu. I'm almost done. So just to mention that why we're seeing continuous interest from government right now to continue doing the work is because COVID-19 was this generation's 9/11, and government needs to continue investing in pandemic preparedness work in order to prevent another COVID from happening, which we're not exempt from that. Climate change is accelerating the pace of zoonotic infectious disease outbreaks that have pandemic potential. And this is just adding on top and compounding other trends like urbanization, mobility. So our communities on the overall are vulnerable. And it's a no-brainer that a platform like this, a wastewater surveillance platform, can really allow you to understand what's happening in communities more real-time, more preventatively, more fast whenever there's a new thing that we can be worried about and want to start looking for. And finally, there's a big movement, as in many other aspects of our life, towards social equity and representation. And wastewater data undoubtedly is a much better representation of what's happening in our communities, of our community human health, versus just looking at what comes out of the clinic. Because not everybody has insurance. People may have different levels of access, or even culturally, if not financially, to access healthcare services. So this is a way to equalize the data that is coming to inform policy and resource allocation everywhere. I should add that one of, besides, I already spoke about some of the other infectious diseases that we are looking at, but I also want to take a moment to say why we're so passionate about looking at substance use. And that's because we believe that substance use is a public health problem. And today, if we, I talked about meth and opioids, and you can see here a sense of the type of data we can get real time. This is a map with a subset of the sites that we work with. And then the color indicates the type of drug consumption that they have. With the purple, as you can see, are communities that consume lots of morphine, meth, and nicotine, not so much fentanyl. And the blue are communities that use a lot of fentanyl and cocaine. This is a type of data that we can get basically real time. This is coming from people who are alive. The data, the gold standard data that public health departments use to tackle substance use today are to look at overdose deaths, which basically when there's an overdose death, there's a mandatory autopsy, which then will do a toxicology analysis to understand which substances resulted in the overdose. But it's estimated that less than 1% of people who suffer from substance use disorder will die of an overdose. So that means that data is not only super lagging, retroactive, stale data, but also it doesn't capture over 99% of people who are alive and can get help. And wastewater presents the only opportunity to collect consumption data from people who are alive to understand what programs to bring where. So we're super passionate about this application, and we are engaged with folks from public health departments, the White House, in order to bring this, similar to what we've done with COVID, bring this level of data collection also for substance use. And just to give an example of how this data can make a real difference, this is the town of Cary in North Carolina was our very first city that we work with before, and this was before COVID, this was 2018. We were looking at the opioid use in their town. And it was such an astounding success, the pilot that we did with them. We collected data from 10 different neighborhoods in their town. And we had two main findings through our platform. Number one, prescription opioids were the dominant type of opioid that was consumed in this town. And secondly, prescription opioid use was pretty much widespread in every neighborhood of the town, which painted a very different picture from what the town knew just looking at overdoses. There seems to be a hotspot in the downtown area of the town, and our data actually showed that this was basically everywhere. So with those two pieces of information, the town designed a program. They designed a safe collection program, as well as a bunch of presentations and interviews to basically give dissemination to the data, to the findings, and they decreased overdoses by 40%, which was the very first decrease in almost a decade, and it was a meaningful decrease as well. According to the NIH, their blue sky in the sky, pie in the sky kind of objective for opioid use is to decrease by 40% nationwide. So that's all that I had, and now I would love to jump into the Q&A session. Thank you.
Thanks, Mariana. Wow, that's an amazing and inspiring talk. So we already have a bunch of questions in the chat. So I'll start reading those. Meanwhile, if there are questions in the room, Kim will be moderating those. And also, if you want to speak online, you can raise your hand and I will call on you and you can unmute yourself for your question. So let me just start reading some of the questions in the chat. So the first one from Renata Kathu is, how do you actually collect a sample?
So there's two go up. One is to go to a wastewater treatment plant, and wastewater treatment plants already have equipment, staff, and workflows in order to collect what it's called a 24-hour composite sample. That's a type of sample that we use grab sample anymore, because with a 24 composite sample, you really get a good snapshot of the entire population, because we assume that everybody's using the toilet at least once in a day. So with wastewater treatment plants, kind of that's the easiest use case, and kind of most of the sites that you saw on the map are wastewater treatment plants, and there are 16,000 of them in the US that capture about 80% of the US population. The other way to collect the sample is where we want it, let's say, from a manhole or from a specific building. So in that situation, you need to bring basically like a sampler to the location and deploy it for 24 hours. And then once it's collected, that sample is sent to our lab for analysis. In both situations, we have a shipping kit where the person who is collecting sends back to our lab like a few tubes of that sample for our analysis.
All right, thank you. Also, just to be clear, for those who want to raise hand to ask questions, I'm talking about using the reaction in the Zoom functions at the bottom of your Zoom screen, because if you're physically raising your hand, there are so many on screen, I won't necessarily see it. So that's just a little technical aside. So there is from Wendy Gilman, 87. She says, looks like sites everywhere but New York. And I was wondering that as well. So what's going on with New York State?
New York State actually does a lot of this work, but there's two situations. One, the work that they do with Biobot, we don't have permission to share their data in our websites. They are very, very sensitive about that. And then part of their work they also do in-house. So in both cases, they are definitely the states so far that we've seen that is the most stringent about just kind of data ownership and kind of their control of their data. They really don't like to give access to their data. So even if we collect some data for them, we don't have the permission to share it in our website. We have a public VES, and yeah, they have never wanted us to share anything from them.
That's interesting and unusual, I think, because it's not like it's individual personal data. Tens of millions of people live in New York.
Yeah, no, it's been the outlier. I mean, there's never been any sort of maybe hopefully one day, if this continues to be so standard, maybe one day they will join because they may be the only gap. But so far it's been 2 years, and yeah, they are not budging.
Okay, that's interesting. We have a few comments. I'll read them out loud. This one's from Anya. It says, Mariana, it's so wonderful to hear your story. Thank you for the work that you were doing and for sharing the journey. There are so many parallels with ESR's journey: perseverance, innovation, and breaking new ground to create new fields of study and practice. And D. Class of 2010, who's calling in from Cambridge, says, this is so inspiring to hear. It really helps to hear research commercialization success led by fellow alumni with research expertise, how you created your research direction, how you found the co-founder, how you decided to create a company versus continuing the research. The vision and conviction are inspiring. Thank you for sharing your journey, Mariana.
Thank you. That's very kind.
Okay, we're going to see if there's anyone in the room who has a question.
So we need to use the mic so that everyone can hear us. Is there anyone who has a question that's here that would like to ask that question? I'll bring you the mic. Julie, go ahead and go on. And if anyone wants a question, they can let me know. Thanks.
All right. Meredith Warshaw, class of 79, is calling in from Newton. She says, as someone who works with infectious disease data, I really appreciate your work, and I'm grateful that you got started on your research early enough to be available for the pandemic. I would second that as well. Very fortuitous that you were already in place. Betsy Salkind, 86, says this presentation is wonderful. Will it be available for those who could not attend today? I'm gonna give that one to Kim.
We are recording today, so we'll make this available after the fact.
Right. Natalie Lorenz, class of 84, says this is so exciting as the next way to ramp up testing and intelligence about community infection and substance use. Is there a business case for the federal, state, and city governments where you can achieve a lowest cost per capital per day or week or month that clearly shows this is superior to individual testing, you know, requiring time, kits, processing, etc.?
Yeah, absolutely. I think that the clinical testing will always be needed in order to basically just for managing specific patients, right? Like the clinical data starts with somebody feeling really sick and then a doctor deciding they need to get tested in the hospital. So I think that will always be there. But the big advantage and ROI of wastewater is that the data doesn't get biased by the availability of tests or current programming or like the people's own, feeling that they need to go get tested. So for times where we are not in a pandemic, I think it is probably the only data that you have. And then when we are in a pandemic, it complements, it gives a leading indicator data. So I think that, yeah, they are complementary in my view. I think they will never replace the clinical one, but yeah.
All right, we have a comment from Rosalie who says, as an epidemiologist who has worked on ways to conduct disease and adverse event surveillance, I've been excited about the methodologic rigor of wastewater surveillance throughout the COVID pandemic. I sure hope the surveillance program continues, and I'm excited to see the opportunities for expansion to other diseases and illnesses. And
Julie, we have a question here in the room.
all right, go ahead. We have several more online, so there's no shortage.
Hi, Sally Harvey Johnston, 71 Civil Engineering. I was watching this and thinking sewage systems only cover a portion of the country. So I just looked it up, and 20% of the country is on private wastewater. So at first I was worried we're missing a large percentage of the population, but maybe not as much as I might have guessed. Thank you, it's great.
Thank you. Yes, exactly. It's about 20%. And for now, yeah, I think we are okay with that 20% not being represented. And hopefully one day we have more coverage, not just domestically, but also in other countries where usually the coverage is more like the other way. Like 10 to 20% population are covered by sewer infrastructure.
All right, I'm going to skip ahead to because some people put in more than one question, and not that we don't want them, but I want to give other people a chance. Sue Wen Kuo, calling in from class of 73, calling in from California, says, did you get involved in the New York discovery of polio outbreak recently? And she also thanks you for your presentation.
Thank you. We aren't involved in that work yet. Interestingly, polio is the one use case of wastewater that has a lot of history and a lot of success kind of already in place. So the CDC, a different we work with, by the way, a different group, they already have the team to be able to test clinical and wastewater samples for polio. And they are the only team that is collecting that data for now. They actually even have some regulation in place that basically if Biobot wanted to test for polio right now, we can't, because there's like some regulatory work that needs to be put in place. And we're right now working with that team to try to lower the regulation so that we can also do polio. So that regulation does feel a little bit like outdated for wastewater. But yeah, that's the CDC, and it's definitely an interesting application. We imagine that again, if the conversations continue to go well as they are, we'll be in position to also offer that type of testing in the future, and will be very relevant, especially for communities that have low rates of vaccination for polio and that are at risk of having an outbreak.
Thank you. All right, Magda says, what substances is Biobot looking to detect in the future, and how can angel investors invest in Biobot? That's Magdalena Luca, class of 1993 and 1996.
So this upcoming year, we're going to be building a bunch of new infectious diseases. So influenza and RSV on the respiratory side. We're going to be launching gonorrhea as a sexually transmitted disease. We're going to be launching norovirus and C. diff, kind of gastrointestinal diseases. And then we're going to keep a bit of bandwidth in order to respond to emerging pathogens, like the things that nobody is expecting right now, but that it may become a priority. We see that all the time. Like this year, it was monkeypox. Like nobody was planning to have a monkeypox outbreak, right? So it's good to have that bit of a bandwidth. On the substance side, we're going to be adding right now xylazine, which is a horse tranquilizer that is getting more and more popular for diversion and misuse. And then on the wellness side, we're going to be also launching the analysis of cortisol and other stress hormones to get a sense of like mental health and stress levels in populations. There's a lot of interest in that one, especially since mental health has just suffered so much, coming out of the pandemic, I think for just like everybody. In terms of angel investing, definitely send me an email because we are raising a Series B. So I'm always happy to chat with angels for sure.
And her email address is in the Alumni Association directory. You can also send me an email. I can forward it to you if needed. All right, Sharon Moran asks, class of 89 asks, has your company worked in Pakistan at all?
No, not yet. So internationally, we've worked in Canada, in Mexico, Ecuador, Uruguay, and right now we're also going to start working in Lithuania. So we have ways to go there. But yeah, if you have any contacts, we're definitely at that point of just wanting to go through our network to start working in other countries as well.
All right, and Dee Yi, class of 10, has another question. She says, may I ask about your startup journey? You had a vision that's grounded with years of academic research and expertise, facing the general startup expectation to fundraise based on specific product market fit. I was wondering, how did you navigate the early stage of the startup fundraising when you first started out?
Oh, I know. Yeah, I would say probably it was easy and not. The MIT brand definitely helped a lot and opened a lot of doors for us, followed also by the Y Combinator brand. I think through that is that we created kind of that first network of investors to get access to. Definitely my co-founder Nusha and I and our company break the mold. So like, 2 women co-founders, immigrants, very technical, and then a company that was saying we're going to be selling to government and something on public health. We definitely had the expected responses of, oh, this sounds like a nonprofit, or like, is this really a company, and how are you going to make money? And the beginning, the first few years were really hard because startups are hard, and in our case, it's like we were playing it on like hard mode, trying to sell to government agencies on something that they had never heard about before. But we persevered, and then when we saw the opportunity to bring attention to wastewater via the COVID pandemic, that's what we did. I think that sometimes investors or people, we like to say that, oh, of course you were doing well during COVID. it was COVID, it was a public health emergency, and you're a public health company. But I feel like people forget that was not necessarily the case. Like, we decided to go and then start doing work in COVID, even though our work to date had been mostly on opioids. We had to quickly pivot and build this entirely new product and capability at the company. And it was in the middle of all of this uncertainty where, if you guys remember, like the first 6 months of COVID, the official message from government was that this was going to go away really quickly. So it was actually, there was nothing guaranteed that this would be an important moment for us, but we were there and we kept pushing and evangelizing and bringing attention, capturing the attention of the public and governments that this is important. And obviously now that's opened the market for us in a way that was not there before COVID, but it was through our active work. It was not just like, that fell from the sky. So I would say in the beginning, very hard, but now obviously like easier because we have a lot of traction, but investors still have the question mark if this is just going to go away once people stop caring about COVID or something. And it's just like, no, we don't think so, but there's always going to be like that type of questions with investors, of course, but I would say easy and not. I also, it's happened. So I think we're fortunate.
Well, thank you, Mariana. We are currently out of time for questions. I will be introducing Colleen Smith, and then there will be online networking and in-person networking so people can chat. So first, I would like to introduce Colleen Smith, who is the AMITA archivist, and she is going to tell you about one of the projects we'll be launching next year to celebrate the 150th anniversary of Ellen Swallow Richards' graduation from MIT.
Thank you, Julie, and thank you, Mariana. What your work is near and dear to my heart. My father was a waste and water management engineer, so these were our dinner conversations when he'd say, "What did you do for work?" and what have you been looking at? So I liked it too from this perspective. To let you know, as many of you know, in 2023, we'll be celebrating the 150th anniversary of Ellen Swallow Richards' graduation from MIT. In the year to come, we hope to bring you more speakers like Mariana, who are building upon the work that Ellen began and going beyond, what Ellen started. We're also, as AMITA, we're working on a set of In Her Footsteps walking tours. The door opens, will be centered in Boston, and will feature Ellen Swallow Richards' MIT experience. What was it like for her as a student? What are the types of connections she made as friends? What were her relationships with the larger community? Through the Open Door starts in 1916, and it highlights the spaces central to women's history and experiences within the current MIT campus. We're going to look at athletics for women, housing for women, and my fun thing that I just found out is I discovered the two women who were the first to hold office, a class office in 1913, they were elected to class office. And one of them is Elizabeth Greenleaf Patty, who also helped develop the landscape architecture for our great court. So it's been kind of fun to go on this journey. You think, oh, I'm looking up a landscape architect, and you discover she was one of the first women to be elected to a class office. Each tour will follow a 2 to 3 mile route and be available as an interactive mobile app for both Apple and Android, because I am a split family, so I'm aware that you have to cater to both, as well as a desktop virtual tour. We hope to launch the tours on May 31st, 2023, because that is the official date of Ellen's graduation from MIT. And in both tours, we're going to be talking about the Cheney Room. So I was thrilled to hear that Lauren was going to let anyone who had come to the in-person portion of this program get a tour of the newly renovated Cheney Room. This summer, Lauren closed it down so that they could do some major, much needed remodeling. It had been decades since the Cheney Room had seen any updates. Margaret's picture was moved to safekeeping with the MIT Museum, and it will be coming back to the renovated Cheney space. And if you're interested about what's this festive space behind me, that is one of our vintage Cheney Room images. In Boston, the Cheney Room had been in two places. The first was the Walker Building, where it was originally installed, and at some point, it was moved to one of the engineering buildings. So I'm not sure which building this room exists in. I'll talk to the MIT Museum to see if they have information, but it's decorated for Christmas. So I thought that was just kind of serendipity that I happened to find that this week. And one of the really cool things too about the research is I've had a chance to work with a lot of great people at MIT. Thera Webb, the Women's Initiative, Women at MIT Initiative archivist, plus the MIT Museum crew. And with the opening of the new MIT Museum, they also reissued all of the photos that they'd had on their old Web Museum digital archive. And it's on a new platform, and it's wonderful. Because if any of you have ever used the older system, you'd find redacted images, missing images. And thanks to their upgrade, I was able to find these additional Cheney Room pictures. So that's been a really fun resource. And if you're looking for the best source of, photographic history of MIT, the MIT Museum Digital Collection is just awesome. I'm having kind of a geek fest right now. So, that's what's coming up for next year in 2023. And this weekend, I was actually at the Cheney estate, and I was in the house where Margaret was born, getting to know them and learning more about Margaret Cheney and her mother Edna, Edna and Ellen were instrumental in the fundraising to create the original Cheney Rooms. And the really neat thing is, and in March, the Cheney estate will be launching an exhibit, The Woman of the Cheney Family, and they will be featuring Margaret because she's the only woman who have gone to a technical school. And even though she wasn't able to graduate because she became ill and died, they're actually going to have an exhibit featuring Margaret and the work that she did while she was a child. And then I'm working with them to help connect it to what she did as a student. And we have found a paper she co-wrote with Ellen Richards, which is really very exciting. So, and in March, so they're going to be dedicating that space in March. So I'm going to be back there come first weekend in March. So all set. That's what's coming up in the corner for me.
Great, thanks, Colleen. I also want to mention we have our AMITA mentoring and networking event coming up on January 24th, so please look for that too. And there's a lot of good stuff going on in the chat right now. I'm not going to read it out loud because I want to give people actually a chance to network either in person or online. So now's the time. If you're online, you can turn on your cameras and turn off your mute and talk with each other. There are a lot of people online, so I'm not sure how that's going to work, but hopefully it will. And people in person can network with each other. And I ask that if you're interested in getting a tour of the Cheney Room and you're here today, stick around for another 15 minutes till 4 o'clock and we'll go over and take a look at it. So I'm going to sign off now and talk amongst yourselves. Bye.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.